Hi, my name is Piotr Janas, and I'm going to present the work on long-term effects of RSV infection on lung epithelial cells that was carried out in Professor Jürgen Schwarz's lab at the University of Edinburgh. It has been recognized for many years now that children that suffered from severe RSV bronchiolitis are more likely to develop recurrent wheeze and asthma later in life. However, the mechanism behind this phenomenon remains undetermined. Here, we hypothesized that RSV infection can imprint lung epithelial cells in a lasting manner, in such a way that could potentially contribute to allergic airway disease development later in life. To study this, we employed a BALB-C model of RSV infection to investigate the effect that RSV has on lung epithelial cells 28 days post-inoculation. First, we used cut and run, which allows us to study genome-wide changes in abundance of histone modifications. Here, we looked at two histone modifications, both associated with transcriptional activation. We found more than 170 differentially bound histone modifications 28 days post-RSV in lung epithelial cells. We also used nanostring to look at transcriptional response of these cells and discovered 6 genes that were both transcriptionally active as well as differentially enriched for histone modifications 28 days post-infection. Strikingly, 5 of these genes are involved in the biology of major histocompatibility complexes and antigen processing. We followed this up by confocal microscopy and flow cytometry to see if we could spot any differences in expression levels of MHC1, here in yellow, and MHC2 in green. And indeed, it seemed like both MHC classes were expressed at a higher level 28 days post-infection. When analyzing cut and run and nanostring data, we also noticed several genes related to antigen processing that remain elevated. To test whether the epithelial cells maintain enhanced antigen processing, we fed them fluorescent of albumin and looked at their ability to uptake and process it. Indeed, we observed that both airway and alveolar epithelial cells exhibit enhanced antigen processing if they experienced RSV 28 days earlier. Lastly, taking all this information together, we hypothesized that RSV-experienced epithelial cells will be able to stimulate T cells to a greater degree than control cells. Indeed, we observed that T cells proliferate more, but also express more CD69 activation marker across several T cell generations. These observations suggest that RSV infection results in long-term changes that last past recovery phase, and since MHC is involved in presenting antigens, including allergens, this phenomenon may be implicated in the development of allergic airway disease like asthma. And with that, I wanted to thank all of my co-authors, as well as British Lung Foundation and Bringing Together Consortium for supporting this work. Thank you for watching this video.